DNA fingerprinting. DNA fingerprinting is a technique that shows the genetic makeup of living things. It is a method of finding the difference between the satellite DNA regions in the genome. What is DNA fingerprinting? Satellite DNA regions are stretches of repetitive DNA which do not code for any specific protein. These non-coding sequences form a major chunk of the DNA profile of humans. They depict a high level of polymorphism and are the basis of DNA fingerprinting. These genes show a high level of polymorphism in all kinds of tissues, as a result of which they prove to be very useful in forensic studies. Any piece of DNA sample found at a crime scene can be analyzed for the level of polymorphism in the non-coding repetitive sequences. After the DNA profile is traced, it becomes easier to find the criminal by performing the DNA fingerprinting for the suspects. Apart from crime scenes, fingerprinting applications also prove useful in finding the parents of an unclaimed baby by conducting a paternity test on a DNA sample from the baby. DNA fingerprinting steps. Alec Jeffreys developed this technique in which he used satellite DNAs, also called VNTRs, variable number of tandem repeats, as a probe because it showed the high level of polymorphism. Following are the steps involved in DNA fingerprinting. Isolating the DNA. Down pointing arrow. Digesting the DNA with the help of restriction endonuclease enzymes. Down pointing arrow. Separating the digested fragments as per the fragment size by the process of electrophoresis. Down pointing arrow. Blotting the separated fragments onto synthetic membranes like nylon. Down pointing arrow. Hybridizing the fragments using labeled VNTR probes. Down pointing arrow. Analyzing the hybrid fragments using autoradiography. DNA fingerprinting applications. As discussed earlier, the technique of fingerprinting is used for DNA analysis in forensic tests and paternity tests. Apart from these two fields, it is also used in determining the frequency of a particular gene in a population, which gives rise to diversity. In case of the change in gene frequency or genetic drift, fingerprinting can be used to trace the role of this change in evolution.